Dear students, welcome and thank you for tuning in to this video. We are continuing our discussion about proteins interaction with biomaterials. In this video, we're going to talk about the protein folding. We have discussed before that proteins are a sequence of amino acids. If you remember, we have this chain. Each one of these circles here represent one amino acid. And those amino acids are 20. From those 20, we have reaction that will polymerize and create these chains. We have amino acids as individual components. There is a reaction that happens in order to chain them together based on a particular sequence. This sequence is very important because if we have any amino acid misplaced, it means that the protein will be not functional. It's not going to do or perform its function. This sequence of amino acids is actually coming from our DNA, our genetic information. The first formation of amino acids to create this chain is called the primary folding, this one here. And this is an amino acid sequence, it's a long chain, and we don't have any folding. In order for proteins to be functional, they have to form into a specific three-dimensional structure by folding. After the chain is created, then we have something called the secondary folding stage, in which hydrogen bonding between the amine group and the acid group in the polypeptide backbone, positive and negative sides of your amino acids or side chains will start to attract or repulse depending on the charges that they have. Before doing the attraction of the positive and negative, hydrogen bonds are the ones that they create the secondary folding. If you look at this image here, this is our primary folding. So we have our chain. The secondary folding is that these kind of structures that we call them the alpha helix and the beta sheets. Those are here called alpha helix structures, and those are what we call beta sheets. So after getting our chain of amino acids, the amino acids will start forming themselves by hydrogen bonds to create this helical structure, and we represent it in textbooks like this, or a sheet structure where the atoms will be arranged in such a way, and we represent it like this as beta sheets. So this is alpha helix and beta sheets. This is the second folding stage. The third folding stage is a tertiary folding, and from its name, it's a three-dimensional structure. And so the alpha helix and beta sheets start folding on themselves as structures to create a three-dimensional object. And then we have something called quaternary folding, which is a fourth dimensional of the folding. We're not going to get into more details about the tertiary and quaternary folding, but this is a picture of how so this is alpha helix and this is beta sheets. That's how we represent them. And when they fold on themselves, alpha helix and beta sheets, we have something called the tertiary folding. And if you have multiple of alpha helix and beta sheets folding on themselves, we have the quaternary folding. I hope this clarifies the idea that how proteins assemble themselves and fold on themselves. We're not going to say assemble, how they fold on themselves to create three-dimensional structures which are functional inside the human body. Now, any amino acid that is misplaced or should not be there in where it is when we produce or create when the proteins are created, we lose the functionality of the entire protein. So that's very important. Thank you for watching. Until next video. Bye.